Hello! In today's video, we shall learn in detail about chromosomes. By the end of this video, you'll have a clear understanding about the structure and types of chromosomes, karyotype, chromosomal rearrangement, and giant chromosomes. Let us begin with the introduction to chromosomes. Chromosomes are thread-like coiled structures made up of proteins present in the nucleus of each cell and contain basic genetic material deoxyribonucleic acid. Chromosomes provide genetic information for various cellular functions and have the ability of self-replication. They play a vital role in growth, heredity, variation, mutation, reproduction of organisms, and in evolution of life. In eukaryotes, each chromosome is made up of DNA tightly coiled around histone protein. The number of chromosomes varies from species to species, but it remains constant for a particular species. Chromosomes, which are morphologically and genetically identical, are known as homologous chromosomes. Now, let us look into the structure of chromosome. The different parts of a chromosome are as follows. Each chromosome has a primary constriction point called the centromere, consisting of small granules. Chromatids or spindle fibers are attached to the centromere. The chromosomes may be monocentric, dicentric, or polycentric, depending on the number of centromere. Centromere divides the chromosome into two arms. The short arm of the chromosome is referred to as the P arm and the long arm is known as the Q arm. The position of the centromere gives the chromosome a particular shape. The kinetochore is a specialized structure within the primary constriction where the spindle fibers get attached. They possess specific DNA sequences with special proteins bound to them. Besides the primary constriction, the chromosomes possess secondary constriction at any point of the chromosome. They contain genes for the formation of the nucleus and are therefore also known as nucleolar organizer. By the way, if you're getting some value out of this video, please like and share this video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Let us continue with the structure of chromosome. During cell division, each chromosome has two symmetrical structures called sister chromatids joined by centromere. Each chromatid contains DNA, and during anaphase, the chromatids get separated and form chromosomes. Chromonema are thread-like coiled filamentous structures present in the chromosome. They control the size of chromosome and are the carriers of genes. The coiled threads or fibrils may be coiled in different ways. They may be paranemic coils or plectonemic coils. Paranemic coils, the fibrils can be easily separated from their coils. Plectonemic coils, the fibrils cannot be separated easily because the chromosomal threads remain intertwined tightly. The coils may be major coils with 10 to 30 gyres or minor coils with number of gyres more than the major coil. Chromomeres are small bead-shaped coiled chromatin present on chromonema that are linearly arranged along the length of the chromosome. Chromatin is a complex genetic material comprising of DNA, RNA, and associated proteins. Nuclear DNA is highly condensed and wrapped around nuclear protein. At interphase, the chromosomes appear as thin chromatin fibers in the nucleoplasm. During cell division, the chromatin fibers condense and are known as chromosomes. The dark stained condensed region of chromatin, which is highly dense in nature, is known as heterochromatin. It contains tightly packed DNA, which is genetically inactive. The light stained, less densely packed DNA is known as euchromatin. It is genetically active and forms the major portion of the chromosomes. Euchromatin actively participates in the transcription of DNA into messenger RNA. Nucleosomes are the basic unit of chromatin. In eukaryotes, histones are required for the packaging of DNA. DNA is wound around histone proteins to form a nucleosome. 8 molecules of histones, 2 each of H2A, H2B, H3 and H4 get organized to form histone octamer. Histones are positively charged due to the presence of amino acids with basic chains. DNA is negatively charged due to the presence of phosphate groups. The negatively charged DNA is wrapped around the positively charged histone octamer to form nucleosome. A typical nucleosome has a core DNA consisting of 146 base pairs. Adjacent nucleosomes are linked with linker DNA consisting of about 54 base pairs. H1 histone is present in the linker region. Telomere is the tip of the chromosome. It prevents other chromosomal segments to be fused with it. The chromosome may possess round, elongated or knob-like structure known as satellite. The chromosome with the satellite is known as SAT chromosome. Matrix is the jelly-like substance present inside the membrane called pellicle which surrounds the chromosome. Next, let us learn about the types of chromosomes. Based on the number of centromere present in a chromosome, there are various types of chromosomes. Monocentric chromosomes have only one centromere and it forms a narrow constriction. Holocentric chromosomes have no distinct primary constriction. They possess diffused centromere and microtubules which remain attached along the length of a chromosome. Acentric chromosome is a fragment of a chromosome without a centromere. Dicentric chromosome is an abnormal chromosome having 2 centromeres. It can be a result of translocation or fusion of 2 chromosome segments, each with a centromere. Based on the position of the centromere, the chromosomes may be of the following types. In acrocentric chromosome, the centromere is situated near the end of the chromosome, resulting in one arm being very short and the other arm being very long. In submetacentric chromosome, the centromere is situated near the middle region, forming two unequal arms. In metacentric chromosome, the centromere is situated in the middle of the chromosome with two equal arms. In telocentric chromosome, the centromere is situated at the tip of the chromosome. It possesses only one arm. Next, let us try to understand the meaning of karyotype. A karyotype is an individual's complete set of chromosomes in the nucleus of the cell. It is a technique used to study the structure of chromosomes, length of chromosomes, position of centromere, banding pattern, and other physical features. A karyotype describes the chromosomal count of an organism. It helps in identifying chromosomal aberrations. In a diploid organism, autosomal chromosomes are present in two copies. Human karyotype contains 22 pairs of autosomal chromosomes and one pair of sex chromosome. In a karyotype, chromosomes are grouped based on the position of centromere. Karyotype of different organisms can be classified into two groups, symmetrical and asymmetrical karyotype. In symmetrical karyotype, all the chromosomes are of the same length, while in asymmetrical karyotype, there is variation in length of chromosome complement. A karyogram or ideogram is a graphical representation of a karyotype. Chromosomes are organized in pairs, and in each group, the descending order of length of chromosome is maintained. Next, let us study about chromosomal rearrangements. Chromosomal rearrangements occur when a chromosome breaks and reunites, giving rise to different configurations. In a balanced rearrangement, there is no loss or gain of genetic material. Chromosome aberration refers to the structural changes in a chromosome causing variations in the arrangement of genes. Unbalanced chromosome rearrangement may be due to deletion, duplication, inversion, or translocation. Deletion can be considered as a mutation in which a part of a chromosome or a sequence of DNA is deleted during replication. It can occur due to errors in chromosomal crossover during meiosis. Deletion may be terminal, intercalary, or microdeletion. Terminal deletion occurs at the end of a chromosome. Intercalary or interstitial deletion occurs in the interior of a chromosome. In microdeletion, minor deletion takes place. Deletion can cause physical and functional changes in human body and can lead to congenital anomalies. Chromosomal deletion syndrome may occur when parts of chromosome are missing. Disorder in human is the partial deletion of the short arm of chromosome 4, resulting in Wolff-Hirschhorn syndrome or 4P syndrome, which affects several parts of the human body with distinct facial characteristics. Jacobsen syndrome is the terminal 11q deletion disorder in which there is loss or deletion of genetic material from chromosome 11. It results in a number of serious health problems, behavioral problems, and memory loss. Duplication occurs when a portion of the chromosome duplicates or is present in two copies, resulting in extra genetic material. The DNA breaks at different points in the sister chromatids. When the ends get joined to other chromatid, it will result with one chromosome having duplicate genes while deletion takes place in the other. Pallister-Killian syndrome is the result of part of chromosome 12 getting duplicated. It is also known as isochromosome 12p syndrome. It is a rare genetic disorder present at birth. This syndrome may show severe intellectual disability, distinctive facial features, results in seizures and other congenital disorders. An inversion in a chromosome occurs when a segment makes 180-degree rotation and is inserted back within the same chromosome but in reverse orientation. The order of genes is reversed. Inversion may be pericentric or paracentric. In pericentric inversion, breaks occur in both arms of the chromosome and it includes the centromere. In paracentric inversion, the breaks occur in the same arm of the chromosome and does not include the centromere. There is no loss in genetic material in this arrangement. Translocation occurs during meiosis when a segment of a chromosome breaks and gets attached to a different non-homologous chromosome. Reciprocal translocation results due to exchange of genetic material of chromosome segments between two different chromosomes. There is no loss or gain of genetic material and therefore the individual retains a normal phenotype. Robertsonian translocation is caused by exchange of parts between non-homologous chromosomes. It is a chromosomal abnormality. It involves the fusion of two acrocentric chromosomes at their ends. An entire chromosome attaches to another chromosome at the centromere. It may lead to genetic imbalances. Down syndrome 21 trisomy is due to an extra chromosome 21. It shows presence of 3 copies of 21st chromosome instead of homologous pair. Finally, let us learn about the giant chromosomes. In some cells at certain stages, amplification in the gene takes place due to which large-sized giant chromosomes are formed containing large nuclei. Giant chromosomes are of 2 types, Polytene chromosomes and Lampbrush chromosomes. Polytene chromosomes are found in the tissues of the salivary glands of Drosophila. The chromosomes are present with many chromatids arranged parallelly. This is due to non-disjunction of chromosomes during anaphase. The polytene chromosomes are larger in size than other somatic chromosomes. They are formed due to repeated or multiple replication of the chromosomal DNA without nuclear division. The chromoneme of the chromosomes get duplicated, do not separate to form new chromosomes, but remain closely associated with each other. The homologous pairs of chromosomes are closely associated, and this type of association is known as somatic pairing. The Polytene chromosomes show alternating dark and light bands along their length. The region where concentration of chromatin is high appear dark. The chromosome is composed of euchromatin and heterochromatin. Euchromatin portion or the dark bands contain large amount of DNA, small amount of RNA and certain basic proteins. The light bands or interbands are fibrillar, composed of heterochromatin, which contains small amount of DNA, large amount of RNA and acidic proteins. Some dark bands of the chromosome swell up or puff out to form chromosome puffs. Puffing is due to the uncoiling of individual chromomeres in a band. Puffs are related with the synthesis of RNA and proteins. Several processes such as accumulation of acidic proteins, synthesis of messenger RNA takes place during the process of puffing. The chromonema of the Polytene chromosome forms series of loops laterally in the form of rings. These rings are known as BOTANY rings. They are rich in DNA and messenger RNA. Polytene chromosome permits a high level of gene expression. It helps in chromosome mapping and in identification of chromosome mutation. Lampbrush chromosomes are the largest chromosomes and is a characteristic of vertebrate oocytes. These chromosomes are also known as diploidine chromosomes as they are formed during the diploidine stage of prophase 1 of meiosis. Lampbrush chromosome is composed of main axis and lateral loops. The main axis is composed of two bivalent chromosomes or four chromatids held together by chiasmata. It has a row of granules known as chromomeres. The chromonema of chromatids gives out fine loops at the lateral sides. The loops are extended segments of DNA and are rich in RNA and proteins. A large number of messenger RNAs and non-coding RNAs are synthesized on the lateral loops. Lampbrush chromosomes are useful for studying chromosome organization, mapping DNA sequences, gene expression, etc. So today we have learnt in detail about structure and types of chromosomes, karyotype, chromosomal rearrangements, and giant chromosomes. I have some practice questions for you. Please share your answers in the comments section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.